Welcome to week number 10. So this week, what we are dealing with is Chapter 8 again, so talking about microbiology. But this week is going to be a little bit different because this week we're learning how to actually grow our bacteria and microorganisms. So kind of want to touch again, remember that Chapter 8 was microbiology, which is the study of microorganisms, which are those tiny entities that are capable of carrying on a living process. Things like viruses, bacteria, fungi, parasites, all of those. And remember that we look at how they're classified by their shape and growth pattern. So the coccus, or that round shape, the bacillus and rod-shaped, or the spiral, spirillum. Okay? Last week that we talked about a lot of the bacteria information, but what we did not talk about was really how to collect, transport, and process microbiology samples. Okay? So it's important when you're collecting a sample for microbiology that you follow, a couple of precautions. Things like making sure you're wearing the appropriate PPE, making sure that you disinfect specimen containers after you collect them. You want to put the specimen in a transport bag after you collect it. And obviously, as always, wash your hands after collection. The ideal time to really collect the specimen is before antibiotics are given. Reason being is after you give somebody an antibiotic, it kills the microbes that you need to test. And it stops them from growing, so you have nothing to test and find out what that microorganism was. So after you collect a specimen, you'll need to go through the process to identify the infectious microorganism. And generally, this is going to be done under a microscope. So you're going to need to do something called a gram stain. And gram staining is really the process of staining microorganisms that serve as a primary means to identify and classify them. It's important to know, and it's helpful to be able to distinguish which gram you have, because gram positive will appear purple in color, where gram negative will appear pink or red in color. You can also do things called smears, which is where you're going to place your specimen onto a glass slide and then stain it to identify whether it is positive or negative. And the last way to do this is to use what we call a wet mount. And this is done to view organisms really in their living state. And it's typically done to detect the presence of a clue cell. And those clue cells are what they will look like and how they react with certain things. So as this is all being, talked about and what we're talking about, these last couple things that we've talked about refer to a physician's office laboratory, or a POL. But sometimes you have to send things out to what we call a reference lab. And when you send microbiology specimens out to a reference lab, it's in order to identify the bacteria and see how it grows. So in order for bacteria to grow, it's going to need a couple of important things. It's going to need things like oxygen, carbon dioxide, and specific nutrients. And those nutrients could be things such as meat extracts, peptone, mineral salts, calcium, or sodium. So now that you know how they grow, you really need to know what they grow on. Now last week I was talking about culture media, and culture medias can be solid, semi-solid, or liquid. But when we talk about a solid-based culture media, it generally grows in a petri dish, and it's called an agar plate. Okay? An agar is really a gelatin substance in which it's obtained from seaweed, and it comes in a liquid form, and then once heated up, it becomes a solid gelatin substance. Again, what you grow things in is called a petri dish. What you grow them on is called an agar plate. Now, because most frequently you will see the use of petri dishes to grow bacteria, you need to understand how to inoculate them. And what inoculation really means is the process of transferring your microorganisms from your sample to a culture media in order to grow them. So there really are two techniques in order to grow and inoculate a culture media. The first one is called an isolation method. And this is when you spread the specimen over 4 different areas of a plate to establish colonies. So you generally will, so if this is your agar plate, you're going to start at the top, and then you'll turn it, and you'll do the side, and you'll turn it again, and do the side, and do the side. So your middle is actually kept empty. The second technique is called the colony count method, which really involves you inoculating the whole culture plate. Now, because we will be doing this in class this week, I'm not going to talk so much about it, but make sure that you take a look at Chapter 8, kind of review that section. Don't reread it, because rereading will not help you. It'll just confuse you more, and you should have already read it at this point. Alright, so that's kind of just a brief overview for this week. I didn't want to go too in-depth because you've already read it, and we already talked about it last week, so I don't want to keep overloading your brain with it. Please don't forget that you do have a discussion post due on Wednesday, with 2 responses due on Saturday. And because this is week 10 and I'm going to be giving you a written final in class next week, we will actually be doing our final review this week in class. So be prepared for that. We'll do that at the end of class on Thursday, and I look forward to seeing all of you. Have a wonderful week, guys. Bye-bye.